Hi, everybody. As we're going through our discussion of Mendelian genetics and the extensions to Mendelism, chapters 3 and 4 in the textbook, I wanted to take a minute to just discuss the three different methods of arriving at our answers, the three methods that are described in the textbook. And really just to get to the idea that all three methods will do the same thing, and it's up to you which one you choose. Sometimes one is more appropriate or easier to use than the other, but they're all appropriate or all will get you to the same place. So the first method that the book talks about is the Punnett square. This is particularly good for either monohybrid or dihybrid crosses. And it doesn't matter if we're talking about simple Mendelian genetics or codominance or incomplete dominance or any of that kind of stuff, epistasis, it doesn't matter. The point here is that we're able to easily do a cross and find out all of the potential outcomes. We're also going to be able to use this later on when we talk about sex linkage and linked genes in general. It's just that the probabilities, the percentages are going to change a little bit, but the basic method will work. So in terms of a Punnett square kind of a setup, if you're allowed to choose your own letters, I strongly suggest choosing something that's easy to read. I hate using the letter C, for instance, because a capital C and a lowercase c look very similar. O is out in my book. I try not to use V's and things like that. But when we talk about doing a Punnett square, sort of the classic example here is a monohybrid cross between heterozygotes. You see this in Mendel's work as the intercross between F1s in a pure breeding kind of a setup. And that would have been a case where we had a cross between two true breeding parent strains, one that has one trait, one that has the other allele of that trait, rather. They generate an F1 organism like this, and when those F1s are intercrossed, we have the situation we started with over at the left. So with the Punnett square, you're just going to draw basically a table. You're going to put one organism along one side and split whatever allele possibilities you have into different rows. And you're going to put the other organism across the top and do the same kind of a thing. For me, to keep this straight, I oftentimes like to label the corner. And so maybe this was a female crossed with a male. Maybe it was a yellow plant crossed with a green plant, whatever it is. I try to put myself a little bit of a label there just so I can keep it straight. And then you're going to put together those alleles, basically representing the act of reproduction, whatever that means in this species, by bringing the gamete from the left side. I'm putting it in combination with the gamete from the top. And left side and top, left side and top, left side and top. You'll notice in the third box, bottom left, that I wrote the capital A first, just because that's the traditional genetic method, is to write the dominant allele first. So I've done that. Then we get to answer our questions, and we can say, what is the genotypic ratio? Well, we can say I get 1 that's homozygous dominant, I get 2 that's heterozygous, and I get 1 that's homozygous recessive. So 25%, 50%, 25%. I would encourage you to think in terms of fractions, because for the most part, fractions are going to be easier here than percentages, but it doesn't matter in this case. We also have the phenotypic ratio, in which now, for the phenotype, we have to interpret what the particulars are of this type of trait. If this is simple Mendelian genetics, well, then we would combine the homozygous dominant and the heterozygote together to say I have 3 that are displaying the dominant phenotype and 1 that's displaying the recessive phenotype. If this was codominance or incomplete dominance, well, then the phenotypic interpretation will be different. I do have 1 that's homozygous dominant. If we were talking about the M and N blood typing that you see in the Extensions to Mendelism chapter, well, then I would have 1 type M at the top, 2 MN heterozygotes, and 1 type N at the bottom. If this was Snapdragon colors, I'd have some red, pink, and white. If this was rabbits with all of their crazy alleles that they have and their coat color examples, I would have whatever the interpretation of these phenotypes is. So what I want to point out here is that we have the genotypic ratios that can be obtained just by counting up the boxes in the Punnett square. And then we have the phenotypic ratio, or what outcome am I going to actually observe. That has to be interpreted based on the particulars of this system. Punnett squares work just fine also for dihybrid crosses. And so I'll be very creative here and pick a dihybrid cross using the letters A and B. And in this particular case, I'm starting with heterozygotes again. So thinking about that Mendelian dihybrid cross kind of a setup. I'm going to do the same thing, and here I'll label these maybe like I did before, female and male. Maybe that's the case, maybe it's not. It depends on your system. This Punnett square is going to be larger, and the key thing to remember is that every row is going to have something from column A and something from column B, and I have to look at all the possibilities. So just for fun, I'm going to change the top genotype here. I'm going to make that a homozygous dominant for B. Okay? So if I put, again, my female down the side and male across the top, maybe we'll do it that way. The female has 4 possible gamete options. I've got a dominant A, dominant B, dominant A, recessive B, recessive A, dominant B, and recessive. All 4 of those have equal probability. If this is straight up Mendelian genetics, those probabilities will change for some examples later when we get into linkage. For the male, notice I only have 2 possibilities because the B allele, I only have a dominant B to offer. So my 2 possibilities are dominant A, dominant B, and recessive A, dominant B. I could have written those into 4 columns, and I could have had basically duplicated effort here, but there's no reason to do that. The more boxes you have to fill in, the better chance there is of just having a random error that you've miscopied something. We'll do the same thing. We'll bring our letters from the top and our letters from the side together to fill in all of our little boxes. And now we can, get our genotypic and phenotypic ratios just like we did before. And you'll notice as I was going through there, I put the dominant letter first, if there was one. I always put the A's before the B's. That doesn't matter, but I just wanted to be consistent. And so I can go through, well, what's the genotypic ratio? Well, now I have to be very careful, right? I've got my AABB, dominant heterozygotes. I only see one of those, top left. If I look and I try to find my homozygous dominant A along with my heterozygous B, now I've got, let's see, I'll go ahead and start crossing these off. Okay, so I found that one already. I've got one here that matches, and looks like that's it. So I just did that in red. So I've only found one of those. Okay. If I go down a bit further now, heterozygous A, homozygous dominant B, maybe that'll be in green. I've got one of those here. I've got one of those up there. So looks like 2 of those. Right? The genotypic ratio is going to be relatively complicated looking here. And I can go through and count up all the rest. You don't need me to count for you. But what I want to say is that as we go through and we fill out the rest, we're going to have 8 total because I have 8 boxes on my Punnett square. So everything is going to be in terms of eighths, or quarters or halves, depending on what your numbers are, but all basically out of 8. The phenotypic ratio is the more common one to think about here. And so I could say, from a phenotypic standpoint, I have 4 options, right? I have dominant A and dominant B. So dominant A, dominant B phenotype. I've got dominant A, recessive B phenotype, recessive A, dominant B phenotype, and recessive A, recessive B. Those are my only 4 options. That dash means I don't care what's in the second position because I already have established dominance in the first position. Okay, so I'm going to go through and erase my little check mark so we can do this again here. Where are my dominant phenotypes? Well, I've got one there that's dominant A and B, and there, and there, and there, and there, and there, right? All of those have some form of dominant A, dominant B genotype, so they're all going to fit into this top category. I don't have anything that's dominant A recessive B, and that's of course because the one parent had homozygous dominant, so I don't have any of those. I do have recessive A dominant B, two of those guys, and I don't have any in the fourth category. So I've got 6 out of 8 in the one category, 75%, 2 out of 8 in the other category. If this had been a cross of heterozygotes, a dihybrid cross of heterozygotes, you'd have 16 squares, and that's where we get Mendel's famous 9:3 to 3:1 ratio. So that's about the largest that you can do for a Punnett square. That's where the other methods come in handy. And so when we look at our other methods, we have the probability method, and we have the forked line method. Both of these are really probability, and it's just a matter of how you interpret it. So let's look at it this way, and let's say these are particularly good for large crosses. Again, I'm skipping the letter C because it's too hard to tell if I have a capital or a lowercase. So let's pretend that we were doing this. Now, if I were doing this in a Punnett square, I would have 8 gamete possibilities for each one of these. So I'd have an 8x8 table, 64 boxes. That's a lot of writing to do. I would go to probability for this, or to forked line. So I'll do forked line first because that shows us the whole picture. Okay, and what I'm going to do for forked line is I'm going to make little, you can do this for probability too, I'm going to make little tiny Punnett squares for each one of my letters. All right, so if I do A and separately B and separately D, turns out these are all 3 crosses of heterozygotes, so it's a bit redundant, but I can very easily, in the margin of my paper somewhere, do this Punnett square crossing. And just like we saw in the previous slide, these are all 1:2:1 ratios because of the genetics that I picked for this problem. But they might not all be, depending on what's going on in your examples. So having that off to the side, now I can say, look, forked line says, here's where I'm starting. Now look at gene A. Well, there's 2 possibilities. So we'll fork it here. Either A is going to be a dominant phenotype, or it'll be recessive. Now, if you wanted to be doing genotypic probabilities, you can certainly have 3 forks here. You could have a homozygous dominant as one path. You could have the heterozygote as a middle path, and the homozygous recessive as the third path. But usually we use this when we're saying, what's the possibility of having some particular outcome? Well, we know that I have a 3 out of 4 chance of going in the upper path, 1 out of 4 chance of going in the lower path, based on that little tiny Punnett square I did. Now, totally separate to that, I'm going to have a choice or an outcome regarding gene B. So again, B will either be dominant or recessive. Dominant or recessive. And I'll look at my little Punnett square I did for B and decide what the right fractions are to put here. Okay. And then I'll go ahead and I'll do the same thing for D, but now on each path. Right, I'm just moving over a little to give me some room. Oops. Okay. So again, either D is dominant or recessive. Dominant or recessive. And again, this isn't the most exciting example because my probabilities are all the same, but you're getting these probability fractions for each letter, each gene, based on what I see over there on the side, those little Punnett squares. And then what I'm going to do is go along and look at each path. If I follow the topmost path, and I go up on every fork, then I get this scenario where I have dominant A phenotype, dominant B phenotype, dominant D phenotype. And I'm going to erase my little Punnett squares so I have a little room over there. Okay. My probability of going this way is 3 out of 4 for A, 3 out of 4 for B, and 3 out of 4 for D. That's the fraction I had on each one of those forks. So 3 times 3 is 9, times 3 again is 27. 4 times 4 times 4, 64, right? I promised you there'd be 64 boxes. So I have a 27 out of 64 chance of getting an offspring here that's going to have all 3 dominant phenotypes. And if I go down each one of these, I'm going to get the same kind of a setup, right? I'm going to put in the fraction for each one. This goes to the idea of probability, that in order to get that outcome, I have to have that 3 out of 4 chance for A happening and a 3 out of 4 chance for B. Anytime I say the word and, I'm going to multiply. So 3 out of 4 times 3 out of 4 times 1 out of 4, that's going to give me 9/64 now. And if I do the math for each one of these, what I'm going to find out is that the dominant offspring in this particular case is going to be by far the most common, 27 out of 64. I'm going to get a couple more 9/64. It's going to be where I have 2 3/4 going on. Okay. I'm going to get some 3/64. That's where I've got a 1/4, a 1/4, and a 3/4. It doesn't matter what order, right? Mathematically speaking, the math's all the same. And I'm going to get the super rare recessive here. Right. And all of those add up. If I add up the numerators, I'm going to get 64 out of 64. And if I don't, I've done something wrong. Where probability by itself, without the forked line, is super useful. Oops, what am I doing here? I need another page. There we go. Where probability by itself is super useful is when I say I'm going to do something really crazy. Okay, so let's say I'm going to go A, B, D, E, F. You know what, I'm going to actually take advantage of the fact that my F's are, maybe that wasn't my best choice. Okay, heterozygote. You know what, I'm going to make A a heterozygote too. Why reinvent the wheel here, guys? Okay, so heterozygous, but now 5 genes. Well, if I were to do forked line or Punnett square, it's unwieldy. But if I say, what's the probability of the offspring having dominant phenotype for A and for B, but a recessive phenotype for D and E, and a dominant phenotype for F? Well, I can do that in terms of the forked line or Punnett square, but probability is the way to go here. Because what I'm going to do is take each gene separately, right? So little tiny Punnett square for A. In this example, A was a cross of heterozygotes, so it's going to look like this, right? So what's the chance of having an A dominant phenotype? Well, that number is 3 out of 4. If I did a Punnett square for B, it would be exactly the same, right? It's a cross of heterozygotes. If I do a cross for D, again, a cross of heterozygotes, be the same thing, but now I want the recessive. That's the 1 out of 4, the bottom right corner. E is going to be the same thing in this example, and F dominant. Okay, so my probability overall, 3 times 3 times 3 on the top, 27, and 4 times 4 times 4 times 4 times 4. You can see how big this thing would have been if we had done a Punnett square. That's 1,024 possibilities, right? So 4 times 4 times 4 times 4 times 4 along the bottom, 1,024. I don't want to draw 1,024 box Punnett square and then try to find 27 of them in that mess. So this does it for us. We're going to come back to these same 3 topics. Think of these 3 things as tools in your toolbox. So you have the Punnett square, which is a great tool. And you can see I even did, that here for helping me with probability. Probability is great, particularly for very complex crosses. And forked line is a nice, visual way to see how things are going. All right. The fractions aren't always going to be the same as I used here. It depends on the initial crosses, right? In some cases, you'll have halves or quarters or whatever it is. You want to just pay attention to that. Be careful. You're going to see an example when we talk about lethal genes in Chapter 4 that sometimes you have to consider that one organism in that box might not survive. And so you might have thirds in those cases. So pay attention to your fractions, but these three tools are going to form our toolbox that we're going to be using for a lot of the next chapters. So with that, I'm going to say goodbye and good luck. And please let me know if you need anything. Keep up the good work here in chapters 3 and 4. And I will talk to you all again later. Take care.